Everyone, I'm Xiaolu Guo, a postdoc from Professor Alexander Hoffman's lab from UCLA, and I really appreciate this precious opportunity to share my research with you, the modeling the heterogeneous NFkB dynamics of single immune cells. And so immune sentinel cells, such as the dendritic cell or macrophages, they can sense diverse immune threats, such as viruses, bacteria, eukaryotes, or damaged tissue. And this environmental information is transmitted through the signaling pathway. Specifically, all these 1,400 pathogen-associated molecules, they can activate the transcription factor NFkB, and which then activates the downstream immune gene expression to initiate the appropriate immune response, such as the cell intrinsic defenses, local immune response, or systematic immune activation. And these immune responses are required to be appropriate or specific to these immune threats. Otherwise, it may cause pathological consequences such as autoimmune disease. And so Taylor and Adilaya from Hoffman's lab, they developed this mouse line, and with this mouse line, we can track the translocation of NFkB, and then get to the data of the activation of NFkB within single immune cells. And from this video, we can see that the TNF cytokine stimulated cells, the NFkB signal shows oscillation pattern, while the LPS is a bacteria-derived molecule. It's the NFkB signal activates, then deactivates slowly, which shows non-oscillation pattern. And we can already see that the NFkB dynamics is stimulus-specific. And besides the stimulus specificity, we can also see that different cell or within the same condition, they performs different behaviors, shows the heterogeneity. And so this through this developed pipeline, we can track tons of macrophages and get a lot of single macrophage data in responding to 5 representative ligand, which includes the TNF, which is a cytokine, CpG, LPS, and PAM, that's the bacteria-derived molecules, and poly-IC, that's the virus-derived molecule. And these 4 are the pathogen-derived ligands. And we tracked more than 7,000 cells. And here shows the heat map of the data. For each heat map, y-axis is different single cell, and x-axis is the time, and the color represents the nuclear NFkB, which is the activation of the NFkB. And from the data here, we can see again the stimulus response specificity, as the TNF stimulated cells mostly shows oscillation pattern, while the LPS shows a high activation, then gradually deactivates. In addition, within the same condition, for example, the CpG, some cells are highly activated and some cells are not responding at all, shows the cellular heterogeneity. And to measure the specific dynamic features of NFkB signaling, Adilaya and Taylor, they defined and calculated more than 100 NFkB dynamic features, and then use information theory and machine learning approach. They selected 6 informative dynamic features they called signaling codon. This codon can review or capture the stimulus information, and they are speed, duration, early versus late, peak amplitude, total activity, and oscillation. And to understand the underlying mechanism that regulates the stimulus response specific NFkB dynamics, we leverage this well-established ODE model, and which is published recently. It composed of 5 receptor modules. And this receptor module will activate the IKK, and then regulates the NFkB translocation through mediating the IKB alpha's degradation. And the nuclear NFκB is what we measured in experiment. And this ODE is a 52-dimension nonlinear ODE system. And this model can account for one representative cell behavior, as the TNF oscillation pattern can be captured by this model, and the LPS stimulated non-oscillation pattern is also captured. But this single parameterization, it cannot account for the heterogeneous behaviors in the population data, as we shown in the previous heat maps. And so there are two categories of possible sources of the heterogeneity in the data. One is technical noise, and another is biological noise, and which composed of the extrinsic noise, that's the differences in the starting states of cells, and the intrinsic noise, which is the stochasticity of the biochemical reactions. And the signal literature shows that within a short time period, the phenotypic heterogeneity mainly comes from the extrinsic noise. And so we then want to ask, is extrinsic noise sufficient to capture the heterogeneous NFκB dynamics? And to answer that question, we developed a workflow and first, we employed a nonlinear mixed effects model. Within this statistic model for individual level, we use this nonlinear model where the Y represents the experimental measurement, and F is the output of the ODE for each individual cell, where the psi is the parameter for each individual cell, and the epsilon is the error term. And so because all the cells are genetically identical, and also they processed the same way in experiment. So we assume that all these single cell parameters comes from the same distribution. And then we will have the population level statistical model where this individual parameter follows a logit normal distribution. And to solve this statistical model, we applied the SAEM algorithm and then to calculate the MLE for population statistic model, and then calculate the MAP for individual cell statistics model. And after we got the result, we assess the model fitting visually and quantitatively. And here shows the convergence plot. And we can see that the algorithm converges. And here shows the results of our model fitting for highly activated NFκB signaling, model can capture it. And for non or low activation of NFκB, model can also capture that. And besides the activation, the oscillation pattern as well as the non-oscillation pattern as well as different oscillatory pattern can be also captured by the model. And we then put all these 15 conditions together, and here experiment, simulation, experiment, simulation, etc. And overall, we can see that the model fits the experiment really well. Then we calculated the RMSD between the experimental data and the simulation data, and apply this stringent threshold. We calculated the good fitting ratio, and we can see that across these 15 conditions, on average, 80% of fittings are acceptable. So here we show that the model topology combined with the extrinsic noise is sufficient to capture the cellular heterogeneity and stimulus specificity in the NFκB signaling. We then calculated the signaling codon. Recall that the signaling codon are the informative dynamic features of NFκB, and here are the Y-line plots of the distribution of signaling codons, and the red is the model fitting, black is experiment. From here, we can see that TNF stimulated low activation is captured by the model, comparing to the LPS stimulated high activation is also captured by the model. We also check the speed codon, which is how fast the activation occurs. And then the TNF fast activation is captured, while the LPS relatively slow activation is also captured by the model. We then plot all this distribution of all 6 signaling codon across 15 conditions, and we can see overall the model can fit the experiment well. And to quantitatively evaluate fitting goodness of the signaling codon distribution, we calculated the average W distance between the experimental signaling distribution and the model. And we can see for most of these 15 conditions, the model fits really well. Thus, we've shown that the extrinsic noise is sufficient to capture the heterogeneous NFkB dynamics. And then we want to ask, does this ODE model match the performance of macrophages, which means that whether the model can capture the stimulus response specificity. To answer that question, the stimulus response specificity is encoded in the signaling codon. So we use the signaling codon as input to train a machine learning model and predict the stimulation. And for experimental data-derived signaling codons, we can predict the stimulation. And we can see for the model-generated data, we can also, from the signaling codon, we can also predict the stimulation, and actually it does slightly better because of lack of the experimental measurement noise. So far we answer that ODE model matches the performance of macrophages in terms of stimulus response specificity. And then we want to ask, can we use model to do forward prediction and discover new biology? And we made the predictions on dual ligand stimulation, and we can see that the model predicts that the different ligand stimulation, they combined in an integrative manner, which means the dual ligand stimulation shows higher activation than single ligand stimulation. And that's validated by our experiment for the LPS and TNF case. For example, the experiment shows that LPS and TNF stimulation is higher than either TNF or LPS. However, different from our model prediction, for the case of CPG and poly-SC, we can see the dual ligand stimulation is actually lower than the CPG activation alone. And we then go back to the signaling pathway and check detail, and found that the poly-SC and CPG pathway, they're sharing this endosomal transport mechanism. So the limited endosomal transport capacity within the cell can cause the competition between these two pathways. So we build this new model implementing this competition between these two pathways. And the new model predicts that the dual ligand stimulation for CPG and poly-SC is actually lower than CPG activation alone, which is more comparable to experiment. And so we show that this model can be used to discover new biology. And we discover that the computation for endosomal transportation can result in a non-integrative combinatorial signaling. And finally, I want to thank my advisor, Professor Alexander Hoffman, and my collaborator, Dr. Adilaya, and Dr. Sen generated all the experimental data. And I'm happy to take questions.
We can take one quick question.
So in your model, you showed very good performance, like RMSD was very high, but this was all based on the data that you gave in. Is there a way for you to kind of think about like test data, right? Like you train the data and then you try to fit it, and so it is a good model fit, but how does it work on like data that was not trained on? Can it Do you see what I'm saying?
Yeah, yeah.
Can you repeat the question?
So the question is that when we calculate RMSD, because we kind of using this data to train our statistic model, and then we test using RMSD. And the question is that whether that's also the performance is also good even for the data that's not trained. And that's a great question. And actually, when we fit individual so we do generate more data, and that's the so it's for non-trained data we cannot compare individually, because for the trained data we have this match for each experimental individual cells. We can find the corresponding simulation, but for new generated data we cannot do pairwise comparison. So here is the generated data, and so we can only compare at population level, and we can see that the generated data performs similar as the experimental data, that the generated data through the signal codon we can predict its stimulation. And so if that answers your question.